Hi everyone, today we're going to continue our Special Topics and New Technologies module with the first part of a three-part medicinal chemistry crash course by Matthew Perry. Part 1 is going to be an introduction on how to start a project. Matthew Perry is principal research chemist at Astra Zeneca, where he works on projects through all phases from hit identification through to candidate selection. He obtained his bachelor's and PhD from the University of Oxford, where he worked with Professor Baldwin on developing new synthetic methodology using hydrozones. After completing 2 postdocs, he became senior research chemist at Pfizer Pharmaceuticals, where he did medicinal chemistry research, and from there he moved to Astra Zeneca. Today, Matthew will give an introduction to medicinal chemistry. And with that, I hand it over to you, Matthew. Thank you for joining us.
Hello everyone, welcome to the crash course in medicinal chemistry. Now, medicinal chemistry is a pretty large field, and even in a fairly high-level introduction, there's quite a lot to include. So to reduce this to more manageable sizes, I've split the talk into 3 parts. In this first part, I will introduce medicinal chemistry. I'll talk about some fairly fundamental concepts such as what is a drug, what is medicinal chemistry, principles that lie behind that in terms of thinking about modalities, about administration routes, about the targets that drugs are aimed at. I'll try and introduce the drug discovery process, and then in the second part of this talk I'll talk about how to start a medicinal chemistry program, what needs to be in place, and particularly what the role of the medicinal chemist is in that initiation process. And then parts 2 and 3 will cover the actual running of a medicinal chemistry program. Part 2 covering the sort of early part and some basic background information. Part 3 covering the more later stages and some special topics, and finishing with some case studies of some nice drug discovery. So let's go on and start the talk. I'd like to start very fundamentally by considering what is a drug. So a drug can be defined as a chemical substance that causes a change in an organism's physiology or psychology when it is consumed. And the fact that it's a chemical substance tells you that you'll need a chemist to discover it. Drugs operate by modifying the action of some kind of biological target, typically an endogenous biomolecule. Now that target could be a protein, an enzyme, a transporter, a receptor, or it could be a nucleic acid, DNA, RNA, modifying the transcription in some way. The target might belong to the patient themselves, but it could also be part of an infectious agent, so a bacterium or a virus. Right, having considered what a drug is, I'd like now to consider what makes a good medicine, a good drug. And the first criterion for that I'm going to suggest is that a good drug is effective. So that a patient who takes it receives the benefit that they're supposed to. And indeed, during the development of a drug, you'll run clinical trials to demonstrate that it does have an effect that it's supposed to do. Now, effects can come in 2 different categories. Patients go to the doctors with some presenting symptom, and so you can give a drug that relieves that symptom. Simple example, you have a headache, you take some kind of painkiller, the headache goes away. But that's probably just relieving the symptom. An alternative possibility is that you actually resolve the underlying problem. Now that's potentially more difficult, but also potentially more beneficial. Now, alongside your drug being effective, the other key aspect for a drug is that it is safe. And safety and efficacy are probably the 2 things that are really critical for a drug. You're developing it, you want it to be something that patients can take that gives them the benefit they want, but also does so without giving them any harm. And undesired effects can come in 2 different ways. There's a side effects, they're typically known as, and they could come as a result of the target that you are trying to affect through your mechanism. So it might be that the target, whatever you do to your target, does what you want it to do, but it might also do something else. If that's the case, then you may have a problem. But the other possibility is that the side effects come through hitting a different target. Now, this is very common. Pretty much every molecule that's ingested into a patient in some way or other will affect more than one target. But if it's a different target, then maybe in your design discovery process, you can prioritize, modify your molecule in order to maximize the effect at the desired target, minimize the effects at the off target. However, when you're doing your development, you need to be very concerned about any kind of target-driven effects, and particularly to try and identify whether undesired effects are driven by activity at the target or some other target. However, these are not the only criteria for a potential drug. I'd like to suggest that it's important actually that a drug is convenient for the patient. You've got to take this medicine. So what do I mean by convenient? Well, how often are you going to take that as a patient? If it's an antibiotic, I guess most people have taken antibiotics during their life, you might end up taking that 4 times a day. Now, if you're taking a course of antibiotics for perhaps a week, that's not too much of a problem. If you're going to take that for several years, taking it 4 times a day is not very convenient. So frequency of dosing can be an important factor. Another case is how big is the pill convenient? There's a kind of joking expression that people use as a horse pill. If your molecule is not very potent, then the patient has to take a lot of drug. Typically, at least if it's a pill, it's a big dose. That's hard to swallow. That's not very convenient. So, something to be considered. And then how to take it. I've kind of assumed in what I've said up till now that it's a pill, but perhaps it isn't a pill. Perhaps you're treating a skin condition, so you might want a cream. Actually, that might be more effective, easier to apply. That's something for the patient. Or perhaps you're trying to treat the lung. Many people who are asthmatics will be familiar with inhalers, administering drugs direct to the lung. So we can think in our design of our drug, how is it going to be taken? And there's one final category of what makes a good drug, which is that it is patentable. And the reason why this is important is that in order for a drug, something that's discovered in a laboratory, to become a product on the market, it has to go through a lot of clinical trials. I'll come on to clinical trials a little bit later, but those are expensive. They take a long time. Somebody is going to have to pay for that. And the usual way that that is done is through an organisation, a company typically, putting up and spending the money to run those clinical trials with the view that should the product be good, the patent will enable them to sell it under a monopoly for a limited period of time to recoup the costs and make some profit. So patentability, important for a new drug. Right, having considered what makes a good drug, let's have a think about what medicinal chemistry is. So I define medicinal chemistry as the science of identifying a molecule as a potential drug. And medicinal chemistry is a process whereby series of molecules are designed, synthesized, tested and analyzed in iterative cycles to identify an optimized molecule that meets objective criteria for development as a drug. So in order to get your new drug molecule, your potential new drug molecule, you'll have to make a lot of compounds. And so the medicinal chemist is involved, first of all, in the decision about what to make, the design process. So somebody, and that's the medicinal chemist, or medicinal chemists in a team, have to say, we are going to make these molecules. And you come to that by some kind of rational process. Then you need to go into the lab and actually make those molecules that you've designed. That's a very important part of medicinal chemistry. However, this talk is not focused on the synthesis aspects of medchem, so I won't talk further about it. Once you've made your compounds, they need to be tested. and you'll test them, and this is generally not done by the medicinal chemist, but the medicinal chemist will probably choose which tests are carried out. So you will have physicochemical tests that understand, to gain understanding of the physical properties of your molecule. You'll test it in various biological assays and potentially even models, disease models, to understand how does it perform. And you'll also want to understand its metabolism. So the testing is very important. And then once you've got those test results, as the medicinal chemists and the design team, you go away and you analyze the results and you try and learn things from them. And you use that to develop either a new hypothesis or more likely revise your existing hypothesis that then enables you to design some new molecules that we will then have to go away and make and test and complete this cycle. And over the course of the project, you will go through this cycle many times until finally you have, you hope, a compound that meets the criteria that you set to be necessary in order for that compound to be developed as a drug. Right, I want to think about the key interactions that medicinal chemistry teams are involved in, and particularly the sort of medicinal chemistry lead. So your project probably starts with, it may well start with just one medicinal chemist, but it will develop to being a team of people with one person having the sort of lead responsibility typically. But drugs are not discovered just by medicinal chemists. It's very much a team activity with people from different disciplines and different skills involved. So we'll think about the key interactions. So first up, we have the project core group. These are going to be the people who get together and really drive the project. There'll probably be a project leader, and then you'll have discipline leads for the different functions within the project. So you'll have bioscientists, quite possibly more than one bioscientist involved in this, because bioscience has many different aspects that feed into this. You'll have drug metabolism and pharmacokinetics lead, and you'll probably have a safety lead. And so bioscience, I said, many different parts. There'll be people who develop and run the assays, and these assays will include both screening assays, but also potentially a high throughput assay. But you will also probably have bioscientists who are involved in doing rather more sophisticated disease models, so that the assays for your screening, your high throughput screen, those will be, you hope, simple assays that enable you to get answers quickly and cheaply on a lot of compounds. And then when you've got good compounds, then you can take them into your disease models, probably initially into cellular models, but then finally, or at least later on, into animal models in vivo. So very important that you have good bioscientists. And the key thing with the project is that everybody's talking together and discussing things, because before you go into in vivo models, you'll need to know that DMPK is right. You'll also be worrying about the safety of your molecule. But this core team is not the whole thing. And as medicinal chemists, these two groups here, you'll be part of the design process. Indeed, the design process is typically led by the lead medicinal chemist. And this will involve input from computational chemistry, and drug metabolism is also a very big part. So this group of people here, the medicinal and computational chemists plus drug metabolism, are the key people in compound design. If you have safety issues, the safety lead may well get involved in this process too. But design, very important part of the role of the medicinal chemist in drug discovery. Now, once you've made your molecule, or any molecules, you'll need probably to formulate them. If you're going to administer them to animals in vivo, that may well require some kind of formulation. So you'll need to get involved with formulation scientists. And then later on in your project, assuming that you're being successful and that you've identified one or more potential candidates to become your development compound, you need to think about How are those compounds going to be administered to patients? Are they going to be as a tablet or as an inhaled formulation or what? And what about the actual solid state properties of your drug molecule? Is it going to be stable? And so you need to interact with the formulation teams, and they'll probably then end up with a representative on the project team. But they're not the only ones. Again, as your drug gets closer towards being put into development, you'll need to start thinking about making not just a few milligrams, but hundreds of grams and then kilograms of material. And that's the area of expertise of the process chemists. And so as the drug gets closer to being selected for development, you'll start talking a lot to process chemists, and ultimately the chemistry will be handed over to them, and they may well completely change the route. They'll certainly investigate it in huge levels, great levels of detail. I talked about the importance of patents in terms of what makes a good drug. As medicinal chemist, you will need to liaise with intellectual property colleagues, and you will have to collaborate with them to produce a patent that protects your discovery. I'd like to talk now a little bit about drug modalities. So in the early days of drug discovery, essentially all drugs were small molecules. But over recent years, the numbers and types of molecules that are used as drugs, and indeed more than just molecules, has expanded greatly. And not all of these are part of medicinal chemistry. So first up, what you might typically associate as a drug, what we would call a small molecule. So a molecule, an organic molecule, molecular weight probably under 500, maybe a little bit more than that. That's your classic drug. That's part of the remit of more Chem. But moving beyond that, we've now got drugs that are known as PROTACs and other bifunctional molecules, which have two different moieties joined together by some kind of linker. So like this compound ARV471, that's also a drug. And then we've got peptides, including cyclic peptides. Those are increasingly drugs. That's a specialized subdiscipline, you might say, of medicinal chemistry. So liraglutide is an example. Even RNA can be made synthetically, or RNA analogues can be made synthetically and have been made synthetically as drugs. There are also treatments for diseases that are developed which are proteins. There are antibodies and fusion proteins. There's CRISPR, gene editing technologies, even cell therapy. These are pharmaceutical treatments, but they're not normally regarded as the domain of medicinal chemistry, and I won't say anything about these. Indeed, I won't say very much really further about peptides or RNA either. There is a specialized subcase. So antibody drug conjugates, which is where you have an antibody and then conjugated to it, there is a drug molecule that is typically released once the antibody is taken up into the cell. And the drug conjugate part of that is very much part of medicinal chemistry. And indeed, the linkers attaching those two antibodies are part of is work that is done by medicinal chemists. So beyond the types of drug, we also need to consider routes of administration. So typically when you think of a drug, you think of some kind of tablet taken by mouth. But there are multiple other methods of administration of drugs. There are topical methods of administration. So for lung conditions, inhalation is a really important means of administering drugs. Some drugs are administered to the skin or through the skin, so sort of dermal administration. You can also get rectal, vaginal, ocular administration direct to the eye, or other drugs are administered through some kind of intravenous or suspension, infusion, or maybe an intramuscular injection. So thinking about your drug, you need to think about the route of administration.
Well, I'm going to move on now and discuss the different types of targets that drugs interact with. The largest class of targets for drugs are proteins of various types, and within those, enzymes form a very large class. So drugs can interact with enzymes either by inhibiting them or activating them. Now, inhibition can happen by a drug binding isosterically, that is to say in the active site, or allosterically, that is to say away from the active site. And an example then of an inhibitor of an enzyme is a beta-lactam drug, which inhibits bacterial DD transpeptidases by mimicking the substrate and binding in the isosteric site. A second very large class of peptide target for drugs are membrane receptors, of which G-protein coupled receptors are probably the best known. Drugs that act on receptors can act as agonists, antagonists, or partial agonists. So an agonist is a drug that elicits the same activity as the natural ligand at the target. An antagonist is a drug that blocks it, whilst a partial agonist will produce some but not the full effect. But beyond drugs acting in that way, you can also get drugs that act as allosteric modulators. That is to say, they bind allosterically, so away from the ligand binding site, and then they modulate the effect of the natural ligand in either a positive or a negative way, so enhancing or reducing the effect of the normal ligand. So one example, very well-known example of drugs that act on a membrane receptor are beta agonist drugs. These are the classic sort of blue inhalers used by asthmatics, and these act as agonists at the beta adrenergic receptor to open the airways and improve breathing for somebody suffering an asthmatic attack. Transcription factors form a third category of protein targets for enzymes. So steroid receptors are transcription factors, and so the glucocorticoid receptor is a major class of a steroid target, and so cortisone and other glucocorticoid agonists act on the glucocorticoid receptor and trigger the transcription of a whole series of anti-inflammatory genes. Ion channels form yet another class of drug target. Ion channels are involved in the transport of ions across membranes within the body, and a very interesting class of drugs, I think, are caftors, and these have been developed to restore the function of chloride channels that are mutant. So patients with cystic fibrosis suffer from the symptoms of cystic fibrosis because they have naturally occurring mutations in the chloride channel that make them less effective. And the caftors have been developed to restore that function and thus treat the symptoms of cystic fibrosis. Similar to ion channels, transporters are also involved in transport across membranes, but this time of molecules rather than ions. And so an example of that is the SGLT2 transporter, the sodium glucose transporter, and inhibitors of that, known as gliflozins, are currently some of the biggest selling drugs in the world, and they inhibit this transport of glucose and are prescribed to diabetics. And the final class of protein targets I'll consider are where two proteins interact, and drugs have been developed that can either inhibit or promote these interactions. And so an example here is an interaction between two proteins called BCL2 and BAX, which are involved in the apoptotic process. At least BCL2 is involved in apoptosis, that is to say a form of programmed cell death, and BAX is another protein that is overexpressed in some cancers that binds to BCL2 in an interaction between two proteins that prevents cells from apoptosing, leading to proliferation of the cells, i.e. cancer. Venetoclax is a drug that blocks that interaction and so permits apoptosis to happen and thus prevents the cancer from proliferating.
Moving on, drugs can also interact with RNA. The first thing actually that you can do is you can make drugs that produce protein. So by adding synthetic messenger RNA into cells, you can get cells to produce a protein. And so obviously the very high profile example of that is the SARS-CoV-2 vaccines from BioNTech and Pfizer and also from Moderna. There are mRNAs that produce the spike protein of SARS-CoV-2. This is synthesized in the cell, released, and the body's immune system starts to recognize this and therefore is primed to mount an immune response to SARS-CoV-2. But you can also use mRNA or RNA to block protein synthesis. And so ways that you can do this is you can use antisense RNA that binds to the messenger RNA, particular protein, and it prevents the translation of that RNA into a protein. And so the example of that was a drug called mipomersen that presented the translation of lipoprotein B100, which is a major component of LDL, and was used in patients with familial hypercholesterolemia. And this was dosed by a subcutaneous injection once weekly. However, this drug was withdrawn in 2019, and antisense RNA has perhaps not been quite as successful as it might have been. Another way of blocking protein synthesis is through RNA interference. So the use of small interfering RNA molecules that bind to a complex and actually cleave messenger RNA, specific messenger RNA, in a catalytic manner. And so the drug there, again targeting LDL, is inclisiran that inhibits the translation of PCSK9, a protease that's responsible for LDL levels. This is also dosed by a subcutaneous injection, but this time every 6 months. So going back to the convenience issue, on the downside, it's an injection. On the upside, you only need treatment once every 6 months. I'd like to talk now a little bit about the process of drug discovery and drug development and where medicinal chemistry fits into that. So projects begin typically with some kind of idea, and there'll be a process where that idea, it's usually for a target, is validated through a series of activities that essentially give the organization that's carrying out the work confidence that the target is worth working on. And at the end of that process, you say, yes, we've got something that's worth following up. We'll run a program to identify a lead. And so this is really where MedChem gets involved. You'll have some MedChem involvement in the target validation process, and I'll come to that in a minute. But then the next phase is lead identification, where you do work on the target, trying to find a series of molecules that you think will actually be good enough to get you to the point where you can get a candidate drug. And once you get that series to that stage, you'll then move into the lead optimization stage, and that will take at least 2 years typically to get you to a candidate drug that you can then nominate into development. So everything up to the nomination of the candidate drug is called discovery. Once it's been nominated, then the candidate goes into development, and it will go through, first of all, preclinical development, taking about a year, and then phase 1, phase 2, and phase 3 clinical trials. And depending on the indication and how much risk the project team and the organisation are prepared to take, these can be longer or shorter. So development of drugs for SARS-CoV-2 went a lot faster than the sum of these, partly by running all of these phases in parallel to reduce the risk. And obviously, each of these, there's a certain amount of attrition at the end of each phase, indeed at the end of each phase in discovery too, that some projects don't make it and they don't meet the guidelines and they're stopped. But The goal is to be here where you've got a marketed product that's benefiting patients. In terms of medicinal chemistry, in the early stages of discovery, you've perhaps got probably just one medicinal chemist spending part of their time contributing ideas to the target validation process. In lead identification, you then start actually doing some real chemistry, making compounds, and then once you commit to lead optimization, you'll put a lot of chemical resourcing effort in, typically, to really get down to the best possible compound you can. Once you've got your candidate, now you may carry on, you may be looking for a backup compound with a different profile, or either of the compound or different risks. But otherwise, there may be some medicinal chemistry involvement into the preclinical development phase, but typically the main bulk of medicinal chemistry work ends at the candidate drug. Having introduced the ideas of medicinal chemistry in a program, I'd now like to talk about what happens in that initial phase where we're starting a medicinal chemistry program. What goes on? What does the medicinal chemist do? So during this target validation phase, we're going to validate the target. We'll have to decide on what the appropriate modality is to inhibit or modify that target to address that target. And obviously that has a really big impact on the medicinal chemistry. I mean, you might decide that actually it's best targeted by an antibody, in which case maybe there's not much more the medicinal chemist is going to do, but you might decide you want to take a topical approach or a PROTAC approach, or maybe just a conventional inhibitor or agonist of some kind. Having made those decisions, then you're going to have to identify some chemical starting points in order to get your medicinal chemistry program going. And you're also going to need to be able to run the assays to do the test part of your design, make, test, analyze cycle. So there needs to be an assay cascade put together that enables the testing of the compounds. And in order to run a project, you need to have both chemical starting points and an assay cascade. Otherwise, you're not going to go anywhere. To start, I'd like to think, how do drug discovery projects start? So I say, a new target identified. Well, where does it come from? Well, it can come from the biological literature, fundamental research into disease processes, and there are particularly studies that knock out individual genes or use CRISPR studies to inactivate particular genes. You can also observe people who have some kind of mutant gene that produces an inactive version of a particular protein, and that can give you clues as to the role of that protein and how it might be beneficial when used as a treatment. So if, for example, you find people who have a gene that is not active and they are protected from something undesirable, that can give you a hint that maybe inhibiting that target can be beneficial. And I'll give some examples of that later on. Your new target may come from in-house biological investigations. I'm assuming that you'll be working in an organisation that has chemists and biologists. Your biologists will probably be doing some fundamental biology, and they may come up with ideas for a target. Another way of generating targets is what's known as phenotypic screening. So you take a cell model of a disease typically, and you throw a lot of compounds at it and see if there are any compounds that affect that model in the way that you're looking at. And then if you find that, then there has to be some sort of scientific detective work to work out how are those compounds affecting that effect. And then you've got the basis of saying, okay, you've identified a mechanism that you can then use to start running a target. Another way of looking for new targets is to look at existing drugs and analyze their off-target activity. So I talked early on about drugs having side effects. Well, if you look at those, you might find that a drug that is out there on the market has some kind of side effect that maybe it's not very potent, but it might be something useful. And there's a saying that the best way to find a new drug is to start with an old drug. And then the final way of finding new targets is to look at the patent literature. So what are your competitors publishing on a particular area? What are they looking into? Is that something that we should also be looking at? Anytime you're working on a project, you certainly need to know what the competitive position is and learn from what others who are working in the field are doing. So for a medicinal chemist, reading the patent literature around your target of interest is extremely important. Right, we've chosen a target, now we've got to validate it. So what do we need to do? Well, the biologists will try to build confidence in the target as something that can be used as a therapy for a disease of interest. So they'll run studies in cellular systems, maybe in animal models. What's this got to do with medicinal chemistry, you ask me? Well, as the medicinal chemist, you need to understand the literature in the field, what's been done before, and if at all possible, to identify and get hold of, synthesize tool molecules. These could be small molecules, they could be antisense oligonucleotides that can be used to modulate this target that's been identified. And as the medicinal chemist, it's your role to understand and to explain to your colleagues the strengths and the weaknesses of the tool molecules. And what I mean by that is, how good are they really? So you'll probably need to profile them in assay panels to understand what they actually do. The literature is full of claims that compound X inhibits target Y, or words to that effect. That may well be true. What they usually don't tell you is what else that compound does. And so you need to be sure that the compound you were using to inhibit or modify whatever it is, your target of interest, does that, and you need to know what else it does. Because you need to be sure when you're basing claims on the use of a tool molecule to do something, that the effect you see is driven by the activity you think it is being driven by. So it's good if you haven't got very good quality tool compounds, maybe they have several off-target activities, is to have multiple tools, because if they have different patterns of off-targets, then you can perhaps draw more conclusions. So very important role for the medicinal chemist at this stage. Now, target validation is not finished at the end of the target validation process. You've merely at that stage reached the point where you can say, well, we've got reasonable hope that this will work in a project. And the ultimate target validation will only happen when you run a large-scale clinical trial in patients. That's when you actually know that your drug works in the way you intend it to. And the reason you run those clinical trials is because quite often they don't. But the better you do your target validation early on, the more likely your clinical trial is to give you the result you want.
Moving on then from validation of a target to thinking about how a project might address a target, one of the things the project team needs to do is to decide which modality or modalities they will investigate to address the particular target. And this is an area where I think the role of the medicinal chemist is quite significant. So what you need to do here is you need to really have a good understanding of the biology of the target, of the role in the disease that target plays. You need to consider whereabouts is the target found. And in this sense, I'm thinking, is it intracellular? Is it membrane bound? Is it extracellular? Because intracellular targets require your drug to cross the cell membrane, and that rules out antibodies, for example, typically, because they simply don't cross the cell membranes. And then you need to think about what type of activity do you want your drug to have? So is just inhibiting it or activating it enough? Or do you actually need to remove it? And that could be degrading it, or blocking the synthesis altogether, or increasing it. So if you need to degrade it, then the Protax or some other method of degradation could be used. Alternatively, you could block the synthesis, and this can be done through some kind of oligonucleotide approach, antisense oligonucleotides, silencing RNA, or perhaps you could inhibit an enzyme that's involved in the synthesis of the target. But maybe you want more of it, so you could increase the target levels by adding synthetic messenger RNA or inhibiting the degradation. So there needs to be a lot of thought into what mechanism you can use to achieve the desired effect. But purely scientific factors are not the only ones you need to consider, because you need to think about the cost of the proposed treatment as well. Larger molecules are, on the whole, much more expensive than small molecules. So if you look at the prices even of a biosimilar antibodies, they are typically well over $1,000 a dose, whereas a small molecule drug in the generic sense is perhaps a few cents to $1 or $2, and that sort of generic cost represents, the cost of goods. Obviously, a patented drug will be more expensive than that. So you need to know what the competitive position is in the disease. If this disease already has treatments that are small molecules, then unless your biologic treatment is really offering a step change improvement, it's probably going to be too expensive to get much take up. You also need to think about the number of patients this disease will have and where the treatment's likely to happen. If it's going to be a relatively small number of patients treated in a hospital, then you can go for more invasive, sophisticated treatments. If it's something that's going to be taken by large numbers of patients, taken at home, then a pill or maybe some kind of simple injection is what's required. I've mentioned already that there are options around administration routes for drugs, and that's a really important decision that you're taking in the early stages of a project, is what route are we going to try and administer our target? So again, you need to be thinking, where is the organ or organs in which your target is found? How is your drug going to get there? Do you need systemic exposure? There are drugs in the blood, plasma, and into certain tissues. Do you need to get into the brain or the central nervous system somewhere? Because there are additional requirements on drugs in terms of physicochemical properties in order to get access to the brain. And so that's essential. If you need to do some kind of centrally acting drug acting on a target in the brain, then it needs to be able to cross the blood-brain barrier. Alternatively, if you don't want to get into the brain, perhaps to avoid some activity from either an off-target or even the actual target, but maybe it has a different role if it's in the CNS, then you can design your drug so that it doesn't get a high exposure in the central nervous system. For some organs, topical administration can be advantageous. Lung, skin, eye are obvious examples, and potential advantages of topical administration. You reduce the systemic exposure, so that actually can therefore reduce the likelihood that you'll see toxic effects. You may be able to get a higher local concentration and therefore a greater efficacy. And some tissues are actually very poorly perfused by the general circulation, and so are actually quite hard to treat by systemic administration. And then you also need to consider for your drug, what kind of profile of action do you need? Does it require 24-hour coverage? That is to say, do you need to have drug there the whole time? And do you need to have a rapid onset? Or would something that took effect over a few hours be sufficient, be satisfactory? And that depends on the nature of the condition that you're trying to treat, whether you want sort of immediate effect or actually something that's going to take place and build up an effect over a few days and have a long-lasting effect will be satisfactory.
Right, one of the most important things in terms of starting a chemical program to discover a drug is to have a chemical starting point. So as the medicinal chemist, your role includes trying to dig into the literature and find, are there any publications? Is there anything known in the scientific literature? Could be journals, could be patents, that gives you a starting point that you can work from. You can look at the biology. Is there a natural product or some kind of intracellular messenger that can be used as a starting point? Or are there any reports of off-target activity of a known drug, or maybe even just activity at a closely related target that you could use? And those are all good things, but I have to say that the most common way of finding chemical matter to start a project from is through some form of screening. So you've got screening for small molecules. Typically you'll do a high throughput screen there. That might screen anything from 100,000 to perhaps 5 million compounds as individual compounds typically. Or you might use what are known as DNA encoded libraries, where you can screen at least potentially billions of molecules for affinity. Your DNA screening, you've got tiny amounts of compounds bound to DNA barcodes, and you'll measure whether they bind to a target and then use DNA sequencing to identify, read the code for the molecule and then synthesize it. Another way to go is to use fragments, so very small molecules, to try and identify starting points. That's a sort of specialized subdiscipline of screening. Or maybe if you're looking for peptides, you can use some kind of phage display system to screen, again, very large numbers. But whatever means you use, you need somehow or other to find a chemical starting point. So thinking about a high throughput screen, you can regard a high throughput screen as a funnel. So I'll give some examples on the right hand side of some sort of representative type numbers. So a high throughput screen, you might screen 2 million compounds, and then a certain number of these will come up as active. That is, that they give a response that is greater than a fixed percentage of change of activity of your target in whatever the way it's being measured. For example, that could be enzyme inhibition at a single concentration. Compounds that come up as active will then typically be tested as 3 replicates per compound. At least that's how it works in my organisation. And compounds that are active in 2 or 3 out of those 3 will be regarded as actives. And that might give you, say, 20,000 compounds. That would be a 1% hit rate, which is not unusual. Okay, you've got these. Well, then what are you going to do? Then you can start thinking about doing a dose response curve, because you've got the numbers down to something more manageable. So perhaps you'll get that down to maybe 10,000, give you some kind of reasonable dose response. Well, this is one assay. There are lots of things that could happen here. So it's a good idea to run some kind of counter screen, something similar that will pick up compounds that are just acting in the same way. So maybe that will get rid of perhaps half of those compounds. So you're now down to about 5,000. And then at this point, you can perhaps run some kind of secondary screen, a different assay relating to your target, a bit lower throughput because you've got the numbers down a long way now, and you can validate the activity. So perhaps you're down to about 2,000 compounds at that point. Well, that's still a lot of compounds. And so you'll need to do some other techniques in order to make that manageable. And what's typically done at this point then is to cluster those remaining compounds using chemical similarity. You'll work with a computational chemist usually at this point, and you'll perhaps generate a couple of hundred clusters maybe. There'll be some clusters with a lot of compounds and maybe quite a few compounds that are just singletons. Once your screen has been run, you've got these series of molecules, and then there's a lot of work here for the medicinal chemist. And for each series, these clusters that you've got, you need to check the identity and validate the activity. And one of the big roles of the medicinal chemist here is to really be skeptical about things. Think very carefully about what you've got and check for properties that might give you false positives. So there's an expression known as pains. They're a set of compounds that have been identified as being likely to come up in assays. You can get things that are known as frequent hitters. You can look at other screens that have been run in your organisation. If the same compounds keep coming up time after time, they're probably not active. They're probably just hitting something or other that makes them appear as a false positive. Different kinds of compounds will show up depending upon the screening technology. And basically every screening technology will have some kind of molecules that affect that technology and appear to be active when they're not really. So as the medicinal chemist, really your job is to check out and try to work out which ones of these are worth following up. Once you've got them and you're at least reasonably happy with them, try to find some analogues of those molecules. If it's been run as a high throughput screen, you'll probably find that there are some analogues available in your collection, maybe not. You might have to buy them in commercially, you may even have to make them, but typically, if you can get hold of things that have been made by somebody else before, so much the better. And at that point, once you've tested them, you can then start to say, well, is there the beginning of some structure-activity relationships? Yes, they are. So that is, when I make changes to this molecule, do they all look the same? Are some of them more active than others? If they all look the same, then that's probably not a very good sign. But if you can see compounds where changes in some place give you a bit more potency, changes elsewhere lose potency, well, that's a good sign, because then there's the sense that, yeah, you can probably do something with this, because your initial hit is likely not to be terribly potent and likely to have quite a lot of things wrong with it, and you're going to have to do quite a lot. So you need to have a series that looks like it's tractable to work on. And then you need to look at the properties of the molecule. So by properties, I mean things like lipophilicity, I'll explain this later, solubility, what is it like in terms of metabolism, just simple in vitro metabolism initially. The objective of all this work that you'll be doing is to identify which of these series of molecules you've got are worth spending more time on, and to spend as little time as possible on the series that aren't going to go anywhere.
Okay, so we've got to the point now where we've run our screen and we're getting ready to get the project going. One of the critical parts of that then is to develop the assay cascade. That is to say, the series of different assays of increasing complexity, assays and models, that we will use to take the project forward. And this assay cascade consists of 3 parts. There's a project-specific part related to the target and the disease of interest, and that needs to be developed by the project team. And then in parallel to that, there will be assay cascades for drug metabolism and safety. These are typically in place there, but they're run in parallel. And so with these 3 different cascades, we will use these to decide how we take compounds forward, which compounds we take forward to progress the project. And the choice of the assays that you use in your cascade are very important, because they will lead to what you discover at the end of it. So for the project cascade, you hope that eventually you will have a computational model that can help you understand what it is about the molecules you're making that leads them to interact with your target, and so you can screen things before you've had to put the effort, the time and the resources into making. So if you can get a computational filter, that's really good. Now that might require you to have something like a crystal structure of your target that you can use then to dock in some way, measure the interaction between a computational version of a molecule and the target. So that's where one of the things that your computational chemistry colleague will be heavily involved in developing. And then you'll have some kind of primary in vitro assay. So if you have, for example, an enzyme as your target, your primary in vitro assay could well be an overexpressed version of this enzyme and measuring inhibition of cleavage of perhaps a fluorescently labeled substrate. so that you've got a very simple readout and you can say, does your compound inhibit this? And so you can screen large numbers of compounds quite quickly and decide which ones are interesting. And then beyond that, you'll probably want some kind of cellular assay. So your initial assay is probably not run in a cell, but then some kind of cell line system where you can measure some kind of probably downstream readout from the effect that you're having. So if, for example, it was an enzyme, maybe then this that you were inhibiting, maybe then there would be some kind of transformation that happened or didn't happen in the cell. Perhaps this led to release of calcium in the cell, and you could detect that. But some kind of cellular assay that measures not only whether your compound is effective against the target, but also whether it has that effect in cells. However, any kind of cellular assay like this is somewhat artificial. So ideally, you'd like then to be running the same sort of study, but using, rather than a cell line, actual human tissue. So this then has to be obtained in some way from patients, probably. You might be able to measure it in tissue sourced from healthy volunteers. For example, if your target is in blood or in plasma, then that can be obtained from patients relatively easily. If it's in an organ, that's a bit more difficult, but some kind of human tissue assay helps you relate the effects you're seeing to what you want to achieve in patients. Going beyond human tissue, you'd like to have some kind of animal model. That is to say, where you could administer your compound to some animals over maybe several days and measure an effect that is related to the condition you hope to treat. And probably you might have a relatively simple animal model to use in the first instance, and then perhaps a rather more sophisticated disease model, maybe in a different species of animal, perhaps a longer lasting model. And the choice of these assays and models is absolutely critical in order for you to be able to take your project forward and to have confidence that, having done so and identified a compound, that it's actually relevant for the condition you're trying to treat in patients. So I mentioned the assay consists of 3 parts. So in parallel to your project assays, you will also be putting your compound through drug metabolism assays, and you'll start almost certainly by running a whole series of calculations on molecules you're thinking of designing to see how they're likely to behave in terms of metabolic properties. So you can calculate physicochemical properties that will impact the metabolism of the compound. And really important to do that, save yourself from wasting a whole lot of time and effort to make a compound that actually is going to be cleared really rapidly as soon as you put it near any kind of human system. And then you'll typically have some kind of primary in vitro assays. These are things like liver enzyme preparations, human liver microsomes, so enzymes from human liver, and perhaps wrapped hepatocytes. So hepatocytes are actual liver cells. You might use wrapped microsomes, but typically 2 sorts of species might well be run there. And then potentially you can run all or many of your compounds through those assays. And then as you get more interested in compounds, you'll run them into some more advanced, more expensive, more complex assays. So things like human hepatocytes. CACO is a cancer cell line that's used to measure permeability. Representative, so it's a cancer colon cell line, so it represents permeability from gut cells and gives you an indication of whether your drug is likely to be absorbed if you're trying to administer it orally. And then at some point after all of that, you will take your compound. If it looks good in these assays and in these assays, you'll take it into some kind of in vivo pharmacokinetic model, which is typically the rat, but it does depend on what your primary animal model is. If your primary model is going to be run in mouse, then you might well want to run your in vitro PK also in mouse. And the idea of this is to understand, well, if we administer it to a mouse, is it going to be there for long enough that we might see the effect that we want to see? And then as your compound progresses, you may need to go into other species. So for example, a dog or a pig pharmacokinetic study. And then the third cascade that you have is a safety cascade. So safety screens, as it were, what other effects might your drug have apart from the target you're trying to inhibit? So all of this will tell you a lot about what you want it to do, but what else will it do? And again, there are now quite a lot of in silico models, so you can predict quite a lot. Now, it may be that for the purposes of developing structure-activity relationships, you make some compounds and test them in your early models that will have quite a lot of off-target effects. That doesn't matter. At that point, you're just trying to learn things. But as you want to take your compound forward into more advanced assays and towards progress your project, you will need to have compounds that are selective. So I'd say you go from initial in silico screening, and then there are various levels of complexity. So there are large panels of targets that are commonly hit by drugs that you can send molecules for screening against, and it's a decision of the project team, and often the chemist and the safety representative have a big part in making these decisions about which compounds you send forward and which activities you look at. And then again, as the compounds progress further, you'll go into more sophisticated safety screening. So for example, looking at genotoxicity. And then finally, as you start getting closer to selecting a compound to maybe go into development, you'll start doing really extensive safety studies. And ultimately, you need to convince regulators that your compound is safe before it's administered to any kind of volunteer or a patient in a clinical trial. Right, you're finally ready now to move forward. You've put everything in place. You've validated your target, you have a project plan, you've selected a modality, you've agreed an administration route, you've found probably multiple hit series and confirmed their activity. You have assay cascades in place, at least the early parts thereof. You're ready to go to whoever provides the funding and seek funding for the next phase. And so this talk will end here, and in the next talk I'll talk about the early parts of actually running a medicinal chemistry program.
Right, well, we've come to the end of part 1. Thank you for watching up till now. In part 2, I will go on to talk about what happens in the lead identification, lead optimization stages of drug discovery, and give you some examples of projects and talk about how these have been applied. So look forward to hearing you then. Bye.
Thank you, Matthew, for a great talk. We'll continue the medicinal chemistry crash course in our next lecture as well, so be sure to tune in for that. See you guys next time.